MHC class II molecules are a family of molecules normally found only on antigen-presenting cells such as dendritic cells, mononuclear phagocytes, some endothelial cells, thymic epithelial cells, and B cells. The antigens presented by class II peptides are derived from extracellular proteins. Hence, the MHC class II-dependent pathway of antigen presentation is called the endocytic or exogenous pathway. Loading of a MHC class II molecule occurs by phagocytosis. Extracellular proteins are endocytosed, digested in lysosomes, and the resulting epitopic peptide fragments are loaded onto MHC class II molecules prior to their migration to the cell surface. Structure: like MHC class I molecules, class II molecules are also heterodimers, but in this case consist of two homogeneous peptides, an I plus or minus and I squared chain, both of which are encoded in the MHC. The subdesignation I plus or minus 1, I plus or minus 2, etc. refers to separate domains within the HLA gene. Each domain is usually encoded by a different exon within the gene, and some genes have further domains that encode leader sequences, transmembrane sequences, etc. Because the antigen binding groove of MHC class II molecules is open at both ends while the corresponding groove on class I molecules is closed at each end, the antigens presented by MHC class II molecules are longer, generally between 15 and 24 amino acid residues long. Expression: these molecules are constitutively expressed in professional, immune antigen-presenting cells, but may also be induced on other cells by interferon I cubed. MHC class II is also expressed on group 3 innate lymphoid cells. Reaction to bacteria: because class II MHC is loaded with extracellular proteins, it is mainly concerned with presentation of extracellular pathogens. Class II molecules interact mainly with immune cells, like the T helper cell. The helper T cells then help to trigger an appropriate immune response which may include localized inflammation and swelling due to recruitment of phagocytes or may lead to a full-force antibody immune response due to activation of B cells. Synthesis: during synthesis of class II MHC in the endoplasmic reticulum, the I plus or minus and I squared chains are produced and complexed with a special polypeptide known as the invariant chain. The nascent MHC class II protein in the raphia has its peptide binding cleft blocked by the invariant chain to prevent it from binding cellular peptides or peptides from the endogenous pathway. The invariant chain also facilitates the export of class II MHC from the ear to the Golgi, followed by fusion with a late endosome containing endocytosed, degraded proteins. The invariant chain is then broken down in stages by proteases called cathepsins, leaving only a small fragment known as clip which maintains blockage of the peptide binding cleft on the MHC molecule. An MHC class II-like structure, HLA-DM, facilitates clip removal and allows the binding of peptides with higher affinities. The stable class II MHC is then presented on the cell surface. Genes. Pathways controlling MHC class II antigen presentation. Pathway, PSD4, ARL14/ARF7, MYO1E . Molecules involved: several molecules are involved in this pathway. PIK3R2 and PIP5K1A are two kinases that create substrates for PSD4. PSD4 is a GEF that loads ARL14/ARF7 with GTP. ARL14/ARF7 is a small GTP-ASE protein that is selectively expressed in immune cells. This protein is localized within MCHI compartments in immature dendritic cells. ARF7EP is an effector of ARL14/ARF7 that interacts with MYO1E. MYO1E is a protein that controls MHCII compartments with an actin-based mechanism. Pathway: PIK3R2 and PIP5K1A are two kinases that phosphorylates phosphatidylinositol, providing PSD4 with substrates for its GTP loading ability. PSD4 is a guanine exchange factor, loads ARL14/ARF7 with GTP. Subsequently, ARF7EP interacts with MYO1E which binds itself to actin myofibers. Altogether, This complex contributes to maintain MHC-loaded vesicles within the immature dendritic cell, impeding its translocation to cell membrane. See also, cross-presentation. References. External links. Histocompatibility antigens Class II at the U.S. National Library of Medicine. Medical subject headings. MHC Class II genes at the U.S. National Library of Medicine. Medical subject headings. http www.merck.com/sec13ch163a html